The overall goal of this procedure is to generate a genetic mosaic in mouse cerebral cortex by inducing Cre-lox recombination through in utero electroporation.
This method of generating genetic mosaics can help answer key questions in neurobiology, such as determining if a gene is necessary for a given neuronal phenotype in a cell-autonomous manner.
The main advantage of this technique is that the effective Cre-lox recombination system is combined with an equally effective gene delivery technique in utero electroporation.
Though this method is focused on targeting layer 2-3 cortical neurons, it can be used to target neurons in other cortical layers and depths.
When we tried to make genetic mosaics in our brain tissue, we encountered several problems in getting our experimental animals to survive after birth. So we introduced several simplifications and refinements to our technique.
Prepare a pipette with a very long taper and very fine tip, typical of pipettes for embryonic stem cell injection or nuclear transfer, by first inserting the glass capillary into the pipette puller. After fastening the clamps, activate the programmed pulling protocol, creating a taper on the glass capillary. Ensure that the taper length is between 10 and 15 millimeters using a compound light microscope. Next, to break the tip of the pipette, center a single-ply task wipe over a 50 milliliter beaker and stretch it taut with one hand. With the other hand, push the pulled pipette taper side down, perpendicularly through the center of the wipe, causing a clean break in the taper. Check the size of the opening under a compound light microscope, then calibrate the pipette.